So this is going to be covering Chapter 9, and Chapter 9 is going to be focusing on the beginning information on the muscles and our muscular tissue. So we're not doing the naming of all the muscles or the location of all the muscles. This is the overarching concept behind the organization of the muscle tissue itself. So we're going to start with 9.1, an overview of our muscle tissue. So this is a very, very important chapter. And like I've prefaced, this is also a very heavy chapter. So this is only going to cover 9.1 to 9.4, which in itself is a very complex chunk of information. So this is not going to be light and fluffy. It is pretty in depth, and we're going to revisit some topics from chapter 2 and chapter 3. Because when you look at our muscle tissue, it is covering nearly half of our body's mass. So this is a very large system that we are working with. And the overall purpose of kind of what it's doing is moving things. And the way it moves our body, it's moving our skeleton and our bones, is it's doing this by transforming chemical energy into mechanical energy in order to exert force. So we're doing that through that molecule ATP that we talked about, adenosine triphosphate. So we're going to talk a lot about this today. As we're going through, we're going to look at types of muscle tissue, characteristics, and then muscle functions. So to start with, the types of muscle tissue we have, whenever you hear the term myo, mes, or sarco, so anytime you see these three prefixes, this is going to be muscle. So as we go, you'll see these kind of starting a lot of words that we'll talk about, not to get confused with each other, but they all are going to fall into the muscle world. But this is helpful when you're looking at an exam and you see myo versus any of the other prefixes we've talked about already, you know that these are going to live in the muscle world. So anytime you see these, we're talking about muscles. There are 3 types of muscle tissue. So this is a brief overview from chapter 4. Of these 3 types, skeletal, cardiac, and smooth, we're going to focus most on our skeletal system. But one thing that is going to be unique to all of them is that whenever you see the term muscle fiber, it is not necessarily going to be a fiber like the collagen or reticular fiber. This is actually going to be a muscle cell. So whenever you see the term fiber, muscle fiber, we do mean muscle cell. And the main reason is most of your muscles are going to be elongated type cells. So they kind of end up looking a little bit more like a long, skinny fiber. I like to describe them kind of like a straw. So they're very long, they're very skinny. So one straw could be a muscle fiber or a muscle cell. So that's just a terminology we need to look at. So starting with our first type of muscle tissue, we're going to have our skeletal muscle tissue. Okay, sorry, my keyboard is showing up randomly. So when we're talking about skeletal muscle tissue, again, this is going to be the bulk of this chapter. This is packaged into a collection of all of these tissues that's going to be called skeletal muscles, and these are going to be geared to moving our bones, and they're going to be attached to bones and our skin. So these are the longest muscle fibers that we're going to have out of the 3 different types, and these are going to be striated. So these muscle fiber cells are going to be long and skinny and have striations. We'll talk through what makes these striations in section 1.3 and 1.4. But these are going to be striated. These are also considered voluntary, which means you choose to move your skeletal muscle. So you are choosing to move these, use these muscles and move them. So it's considered voluntary, it's consciously controlled. And as these are going to happen, you're going to go through a process where we're going to contract these skeletal muscles. So we'll talk through that. So when you're looking at the comparison of the 3 muscle types, what you need to know is these keywords right here, or these pieces of information right here. That is, skeletal muscle is striated and voluntary. The second type is going to be cardiac muscle. And cardiac muscle is specific to our heart muscle. So this is only going to be found in the heart. Makes up the heart walls. It is also striated. So this type of muscle tissue also has these striations, but it is considered involuntary. So you do not actually choose to have your heart beat. It does it. It knows what it needs to do. It is controlled unconsciously. So cardiac muscle is considered involuntary. Those are the key words for that. And then the third type is going to be smooth muscle. This is going to be briefly covered in the last section in chapter 9. Cardiac muscle won't be covered really until second semester when you're looking at the cardiovascular system. But smooth muscle will be covered briefly, and this is going to be found in the walls of our hollow organs. So things that are like stomach, urinary bladder, airways, anywhere where you're going to have a hollow organ where you may constrict the inside or make the inside larger. This is not striated, so these muscle cells do not have the stripes. But this is also going to be considered involuntary. So you don't actually choose to have your digestive tract or your urinary bladder move things through it. So that smooth muscle is not under the conscious control of you knowing that you want to move it and you making it contract. So it's considered involuntary. So these are the three keywords for our smooth muscle. And here is a lovely comparison. But again, those three keywords, whether or not it's striated or not, whether or not it's voluntary or not, and the type of muscle are the things you need to know. Okay, when we're looking at muscle tissue, there's a couple characteristics that they're all going to have, and these are really important, but they're going to come up as we talk about muscle tissue throughout the rest of the chapter. When we're talking about characteristics and we talk about excitability, what we mean is that your muscle tissues will have the ability to receive stimuli and then have some sort of response to that. So this excitability feature essentially just means that it can pick up a stimuli, and then excited is essentially going to mean that the cell will have a change in its resting membrane potential. It'll be excited, but it's going to be able to respond to some sort of stimuli. Another characteristic is contractility. And contractility is going to be the ability for it to forcibly shorten. So after it's been stimulated, it can go from one length and it can contract to a much shorter length. We're going to talk a lot about this. This is how we actually move our skeleton. Then another characteristic is going to be extensibility. That's a tongue twister. Extensibility is the ability to be stretched. So after we've contracted our muscle, we then need to be able to extend our muscle fibers back. That's the characteristic. And then the last characteristic of our muscle tissue is elasticity. And elasticity is that when it's not contracting and when it's not being extended or stretched, it can always go back to its normal resting length. So it has the ability to go either contracted, extended, but always can come back to its normal resting length because it's elastic. So those are the four characteristics of muscle tissue. The reason we need these characteristics is because of these important muscle functions. So some of the overarching, and you'll notice that these are not specific to skeletal muscle right now. This is going to be a general muscles, and then we're going to go through skeletal muscles specifically. So the overall functions of muscle. The first one is going to be to produce movement. When we're talking about skeletal muscle, we are moving our bones or our skeleton. But when we're talking about smooth muscle, we're going to be moving blood through our system, and we can be pumping blood through our arteries and our veins. But either way, muscle is producing some sort of movement. The second function of muscle, whether it's cardiac, smooth, or skeletal, is going to mainly be focusing on our skeletal for these. It's going to maintain the posture and our body's position. So the ability to stand upright and stay standing upright is because our skeletal muscles are holding our bones in place. The ability to then bend over and not just fall flat on our face is due to our skeletal muscles working and being connected the way they are. The third muscle function is going to be to stabilize our joints. So this goes hand in hand with our posture and our body's positioning, but as we're stabilizing our joints, It's essentially holding that in place. So you don't just have floppy arms, but you are able to maintain a straight arm or a bent arm. It's not just all over the place because you cannot control your elbow joint. So muscles help stabilize the joint in place. And then another cool thing, but we've already briefly talked about this, is they have the ability to generate heat. So when we talked about homeostasis in chapter 1, and we went through a decrease in temperature and how your body will actually shiver as a response in order to generate heat, it'll contract. That is what's going on. So you actually, when your muscles will contract, they will essentially create a energy, which is going to be a heat, and it will heat up the system. So this is why when you shiver, you're actually contracting your muscles in order to get that heat when the temperature is really, really, really cold. So this will make a little bit more sense when we get to how the contraction actually works. Okay, so for 9.2 to I believe 9.8, everything else we're going to talk about is going to be skeletal muscle from this point forward. So that's the world we're going to be living in. So remember, skeletal muscle is striated and it is voluntary. So that's what we're going to live in right now. So when we're talking about skeletal muscle anatomy, we are talking about an organ, and this organ is going to have different tissues, because remember, that's a definition of an organ. There's going to, yes, be the muscle itself. There's going to be nerve and blood supply, and there's going to be connective tissues that are going to be attached and around the actual skeletal muscle itself. So when we're talking about the nerve and blood supply, the things that are going to actually be the gross anatomy of the muscle. So we're going to start with this. This falls under the category of the gross anatomy of your skeletal muscle. Okay, so the first thing for our gross anatomy that we're going to have is we are going to have the nerve and the blood supply. So you will have innervation, which is going to be the nerve that can pick up sensations, and you'll have blood. So you'll have all adequate influx of nutrients, being able to take away waste, everything that you will need. It's a rich blood supply. This is really, really, really important for the nerve, artery, and vein, is that as you actually go through the process of contracting, you will actually use huge amounts of oxygen and nutrients. So we need a lot of blood in order to be able to produce the movements that we are going to talk about. So this is highly, highly, highly important for our skeletal muscles to have nerve and blood supply. The next thing we're going to have is going to be connective tissue sheaths. So basically, this is how we are connecting our muscles to our skeleton. So these connective tissue sheaths are essentially going to be the hammock surrounding the actual muscle tissue. So each skeletal muscle, and also each muscle fiber, so every individual muscle cell, is going to be covered in connective tissue. So that means that these sheaths will go from internal and they will go all the way to external. So this particular right here is going out to in. So the function of these connective tissue sheaths is to support the actual muscle itself and to support the cell. So this would be to support the muscle fiber, also known as the muscle cell, and also to hold together the muscle. So there are 3 layers. These are going to sound kind of familiar because we've talked a little bit about other things that sound like this. So if this is my muscle fiber, remember he's a straw, and our muscle fiber is actually going to be surrounded by one type of connective tissue sheath, and this will be connected by endomysium. Endomysium is going to be the most internal one that is around each muscle fiber. You put this one straw together, so here is my muscle fiber right here, and he is surrounded in endomysium. That's the little clear surrounding around him. So this one straw, if you put this one straw in a bunch of other straws all the way together, you essentially have created a packet of straws. That is going to be something called a fascicle. So we've got a muscle fiber, which is our actual straw or our actual cell. We put those together and we are going to get a fascicle, which is a group of straws together or a group of muscle cells together. So all of these are going to have a type of connective tissue around it. That is going to be the perimysium. So we're going to have saran wrap, essentially, connective tissue around each individual straw. We're going to then put a bunch of straws together and put saran wrap around that group of straws. So that would be our perimysium that's surrounding a fascicle. And if I put a bunch of straws together and you put a bunch of straws together and someone else puts a bunch of straws together and then we put all of those together, essentially then you're going to have the actual muscle itself. So when you're looking at it like this is actually going to be what people are considering your muscle. It's going to be a bunch of different selections of fascicles put together. So these muscles are then going to be completely surrounded in connective tissue, and that's the one that's most external, which is going to be your epimysium. Epi means upon, endo means in, and peri kind of means middle. So that's one good way to remember it. So now you can see here how we have the muscle fiber, which is this guy right here, and he is surrounded in an endomysium. You put that muscle fiber in a bunch of other ones, and you put them all together, and this right here is going to be my fascicle, and that is surrounded with this clear layer of perimysium. You put a fascicle and a lot of these fascicles together, so that's what all of these individual guys are, and then you surround that with a connective tissue, and that is perimysium, oh sorry, that's epimysium, that is then surrounding your actual muscle. So now we kind of have this saran wrap that is connected around our entire muscle. Next, we're going to actually use those connections, and we have to figure out how to attach the muscle to bone. So the way we're going to attach this muscle to bone is going to be actually pretty cool. So muscles will span joints and attach to bones. So they will always attach in at least two places. So we briefly talked about this in the chapter before with joints. Insertion is the movable bone. Origin is the immovable or the immobile bone. So if I'm going to have a sweet little 4-year-old and he is going to essentially, here's his elbow and here's his hand, here's his elbow, and here's his hand. So we've got our humerus, our elbow, and then our radius and our ulna. And then you ask him to flex. Essentially what he is going to do is he is going to move that arm closer in, and he is going to flex. The way he does this and how it actually works is because the muscle that is attached here at his shoulder is also going to be attached here on his arm, attached here at his shoulder, and attached here at his arm. So when he flexes, it goes from here at his shoulder, here at his arm, on both. So when that actually happens, this bone right here, the one that has not moved, is considered the origin. So this one is going to be the bone that does not move. It is the origin. It's kind of like your place of birth. That never change. Where you originate from will never change. But the bone that was attached to the muscle and then it was moved, that would be the insertion. Where you insert yourself in life has the ability to change. So that's how I like to think about it. So those two terms, insertion and origin, your skeletal muscle will always attach two places, and it will be named for usually where it connects. And then now we're going to second talk about how you actually connect the muscle to the bone. But those are two terms you need to know, insertion and origin. So now that we have these and we know which one is which, how do we actually connect these to the bone? Well, attachments can either be direct or indirect. So remember, here's my straw, here's my fascicle, and here's my actual muscle fiber. My actual muscle. So direct attachments can be direct or indirect. So they can actually be attached directly to something, or they can essentially take this connective tissue that's around it, grab that together, and then that can be directly attached to something. So attachments to things are going to be considered, attachments to bone, not just things, are considered direct or indirect. A direct attachment is your epimysium. And your epimysium is going to be the outside top layer of your muscle fiber. So with a direct attachment, your epimysium is fused to the bone itself, but technically it's fused to the periosteum of bone. It may also be fused to the perichondrium of cartilage, but it's usually going to be bone. So direct. Or you can have indirect. And indirect to me kind of looks like a hammock. And this is where you will have connective tissue. And this connective tissue will extend in a tendon. And then it will attach to a bone. So if you've ever gone camping or used a hammock, here's my skeletal muscle. Around it, I'm going to have this connective tissue. It will actually come together on either side, kind of like a hammock, and that is what then will attach to my bone. So it's not directly connected, it is indirectly connected. So you can see right here, this is going to be an indirect attachment because it's got that connective tissue that extends into a tendon, and that is what's attached to bone. Okay, so the next for 9.3 and 9.4, these are the heaviest focuses for this first half of chapter 9. 9.3, we just did the gross anatomy, so now we're going to do microscopic anatomy. So this is the microscopic anatomy of skeletal muscle fibers. And this is going to be pretty nitty-gritty. The microscopic structure of skeletal muscle fibers. So whenever we're going to talk about our skeletal muscle, remember we are talking about it as a muscle fiber. Whenever we say cell, we mean a muscle fiber. And these are long cylindrical cells that are going to contain multiple nuclei. So I like to just describe them as a straw. So that means that inside your nuclei, inside your cell, you'll have a bunch of nuclei. They will typically be shoved to the sides for reasons we'll talk about in a second. So these are long and they have multiple nuclei. There's a couple new terms. Remember we said anything sarc meant skeletal muscle or meant muscle. So from now on, in this particular chapter, whenever we talk about sarcolemma, sarcolemma is going to be the plasma membrane. So it's not the phospholipid bilayer, it's not the plasma membrane. It is always from this chapter on, going to be called the sarcolemma. And inside that plasma membrane with all of the organelles, we will have a sarcoplasm. So this is our cytoplasm. It has a different feature, a couple different characteristics, and a couple different organelles. So our sarcolemma is our plasma membrane. Our sarcoplasm is going to be our cytoplasm. So those are a couple different terms for this chapter. And then inside our actual cytoplasm, our sarcoplasm, there will be some things that are going to be kind of new modified organelles. And these modified organelles we'll talk about because they're going to have a very specific function. So we're going to have ones that are called myofibrils. That's the one we're going to do first. And then you're going to have sarcoplasmic reticulum. Remember, kind of like the endoplasmic reticulum. This is the sarcoplasmic reticulum. And then something called T-tubules. So these are found inside the sarcoplasm. They are modified new organelles that are found inside our skeletal muscle fibers. So we're going to start with the first modified organelle, a myofibril. This is the first type. When we're talking about a myofibril, what we're talking about is something that is going to be kind of like a rod-like, but very, very, very densely packed thing inside the cell. So there are myofibrils. They make up about 80% of the cell volume. So they are these rod-like elements that are a vast amount of the cell. So what I like to describe these as, if this was going to be my muscle fiber, where we know it's a long straw, and we've got our nuclei that are all the way, we've got multiple nuclei in it. Our myofibrils, I like to describe as toothpicks, which are rod-like kind of structures, and they are shoved inside this straw in a very organized manner. And we're actually going to discover that we have two different types. But we are going to take these essentially toothpick-like structure and shove it into very, 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 very densely packed, so much so that a single muscle fiber can contain up to 1,000 of these. So we're going to take all of these little like toothpick-like structures and put them into our straw in a very organized, specific manner. Those are going to be our myofibrils. Our myofibrils are going to have some very specific features that make them what they are. And these features are going to be striations, sarcomeres, myofilaments, and the molecular composition of the myofilaments. Remember, we said anything myo would also mean muscle. So sarc and muscle. Okay, we're going to talk about these fibril features in a second. We're going to go through all of these, but those are the four things that make it up. So when we're looking at our myofibrils, they are going to have striations. So your myofibril will have striations. This is what makes your skeletal muscle have striations. And these striations are going to be because there are alternating light and dark bands. So these alternating light and dark bands is what gives it a striated appearance. And so when we're talking about a light band, that will also be called an I band. Whereas when we have a dark band, that will be called an A band. So an I band is going to be a light region, and an A band is going to be a dark region. So when we're looking at here's a blown up version of a striation, that would be a dark, that would be a dark, that would be a dark, but then these would be the light. So that's kind of how the structure works in order to give it these striations. The H zone, M line, and Z disc we'll come back to in a second. We're not going to forget those, but right now we're just going to talk about the light and the A bands. Okay, so you can kind of see how we've got the stripes here, alternating dark and light. And then another feature, not just the myofibrils, but another feature is going to be the sarcomere. So this is a very, very, very unique feature, is going to be this sarcomere. So a sarcomere is considered the smallest contractile unit of the muscle fiber. So this is the thing that actually makes the muscle shorten, is going to be this sarcomere. This is very, 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 very important, this sarcomere. And what it's actually going to consist of is it is going to be the area between the Z discs. So you're going to have this lovely little structure that looks like this. This is going to be a Z disc. This is going to be a Z disc. And we're going to talk about a structure that makes up all of the middle. So this whole middle will have something really, really cool. But from a Z disc to Z disc, from here to here, including everything in the middle, This will be your sarcomere. So everything that will contract, it's the smallest contractile, it will be this sarcomere. So you can see down here, there's my little diagonal line I drew. There's my Z disc. There's my Z disc. Again, here's everything in the middle, exactly what I drew over here. So from here, your Z disc to your Z disc, that is your sarcomere. So that's what we're talking about. Now we're going to figure out what the stuff in the middle is and what makes that up. But along your skeletal muscle fibers, you're going to have Z discs all the way down. And those are going to create some of those organization for our myofibrils. Cool. So our next section is actually talking about our myofibrils and their organization. So we're now going to go into our myofilaments. So remember, our myofibrils are the toothpicks. They are the rod-like structure. Our myofilaments is what makes up our myofibers. So within our myofilaments, there are going to be a very orderly arrangement of two types. Actin and myosin are going to be found within the sarcomere. So there are two types of myofilaments. The first type is actin. Actin is going to be also considered the thin filament. So whenever you see actin, it is going to be thin. This one looks like essentially a string of beads. There is one bead string, here's another string of beads, and all the way down my lovely little string, I am going to have these actin protein. So when we're talking about a thin filament of actin, what it is it is going to be two strings of actin-like beads that are going to be strung together like this. So this is our thin or our actin filament. So our actin is going to be the little purple things that I drew. This is the actual protein. So this is going to be your actin. But then what it is actually attached to is it is going to be attached to this little like rope, and that is going to be called your tropomyosin. So the actin protein is attached to the rope that is called the tropomyosin. In order to hold your tropomyosin and your actin in place, all along it, there's going to be these little anchor points that hold everything together. These anchor points are going to be troponin. So on our actin filament, it is a thin filament. It is made up of an actin protein, and it is going to have tropomyosin strands with the actin protein attached, and a troponin molecule is holding it all in place. So that's what this guy looks like. This is our actin or our thin filament. I don't love the way the PowerPoints go in order because they kind of skip around in some of the images. So we'll see this in a couple slides, but I want to draw about it and talk about it here. So then the next type we're going to have is going to be our myosin filaments, myofilaments. And our myosin is also a protein, but it's called the thick filament. So you've got the thin and the thick. This one looks like a golf club. So if I were to take a golf club and put them all together, I'm trying to find a good color here. Essentially, here's my golf club, and then my golf club has this where you actually hit the ball, the head of the golf club. This is your essentially a myosin. You take this one myosin molecule that looks like a golf club, kind of looks like a candy cane as well, and you put a bunch of these together. So here's another one. It's kind of like where you're putting all of these guys in a bag, where you've got all of the golf clubs that have to kind of point out at different directions. So this is your myosin with all of the heads going in one direction, but you can also then have it where the heads go in the other direction. So a myosin filament, which is a thick filament, looks like this. Kind of like golf club together with the golf club heads kind of all pointing in different directions. There's usually about 300 myosin heads per myosin. Okay, so this is what's going on with our thick and our thin filaments, is they look like this. And again, we'll talk about Z-disc, I-band, and all this fun stuff. So a sarcomere cross-section is going to be a thick filament and thin filaments. So here is our sarcomere. So remember this guy right here. Z-disc to Z-disc is our sarcomere. Inside our sarcomere, we're going to have our blue thin filaments and our red thick filaments in a very organized manner. So this goes into a little bit of an overview of what we just talked about with our myofilaments. So our thick, which is going to be our myosin, is going to have a myosin tail and a globular head. And when you put all of these together, it'll get a staggered array of heads pointing in different directions. So here is my golf club example, a little bit late to the game. So here is my myosin head, all looking like this. And remember, they are staggered and on both sides of the tail. So this is what it looks like. That's your thick. Whereas your thin actin filament is going to be made up of a protein, and it is going to be globular actin subunits. And these are going to be linking together on a protein. Tropomyosin and troponin are on our protein. So here's what we drew last time. So here's our strings coming together of our tropomyosin. And then on our tropomyosin, we're going to have our actin protein. So this is what we're talking about with our thin filaments. When we put these together, what we're actually going to have is tropomyosin. We're going to have our actin, our myosin, and our sarcomere all put together into the same concept. So as we do that, at the very, very bottom of this, the one I want you to see with this molecular composition of myofilaments, the only other protein I really want you to focus on is going to be this titin. And this is going to be involved with our thick filaments. It's going to hold it into place. So that's something I want you to notice. So when we're looking at our sarcomere, I'm going to come back up and we're going to draw this guy. So your sarcomere is very, very, very important. So this is the biggest takeaway from section 9.3 is how this whole process is organized. So we're going to blow it up and we're going to draw it. So here is my Z-disc, and he looks like this. So here's my Z-disc to my Z-disc. So this whole guy is my sarcomere. I know I've said it multiple times. Okay, so the way this works is this Z-disc attached to your Z-disc, you will actually have your thin or your actin protein. So attached to your Z-disc is your actin protein. It does not cross all the way to the other side and meet in the middle. But on each Z-disc, on each side, that's why we drew it like this, you will have your actin molecule attached to your Z-disc. Then the next thing you're going to have is you're then going to have that teeny tiny little protein we talked about, which is going to be our titin protein. And that will be connected to the other side of our Z-disc. This will be our titin protein. I always want to call it Tintin. Our titin protein. And we said our titin protein was involved with holding our thick filament in place. So stretching from one side to the other will be your thick filament. Stretching from one side to the other of my titin protein will be my thick filament. But remember, this thick filament is going to have those heads that come out in varying directions. So this is going to be my thick filament that kind of looks like this. But it's your titin protein that is holding it in place. So we've got our Z disc, we've got our actin connected to our Z disc, we have our tin protein, titin protein, holding our thick filament in place. You'll notice your thick filament does not touch the Z disc. The thick filament does not touch the Z disc. Okay, so when you're looking at this, one thing that's holding this whole thing in place is right down the middle of our thick filaments. This will be our M line. So your M line is going to be holding and anchoring. Another way to anchor your thick filaments in place is through this M line. All right, right down the middle. M stands for middle, so that's pretty easy. Okay, so now when you look at it and you see this, we have organized our sarcomere, our actin, I did not actually draw it, but this is our myosin. But how it actually is organized is you will notice that right here in this section, right here, from right here to right here, this part right here is where we are only going to have, so right here to right here, there is only thick filaments. So this section right here is only going to be where you will find our myosin. So this will be thick only. But then what you'll actually have, if that's our thick only, this section right here, it's right next to it, you will have an overlap of, right here, thick and thin. So we've got our red and we have our groove. So then we'll go on the side over here too, all the way to the Z disc. My proportions are a little bit off, but you will have your thick and your thin. Let's go ahead and get this guy back. There we go. So right here, you will have your thick plus your thin. When you have your thick plus your thin, you'll see that on both sides. This right here will be your I band. This will be the light band. It is going to be made up of your thick filament only your myosin. Whereas next to it, you will have your A band, which will be your dark band. And that is made up of both your thick filament and your thin filament. So you'll have that on both sides where you will have your A band. This is how you get the striations of all the way down your skeletal muscle fiber, alternating light band or an I band and a dark band or an A band. Light, dark, I band, A band, I band, A band. It's happening because in the middle of your M line, you are only going to have the thick filament, whereas on either side of it, you then will have thick and thin filaments. And this is how you get the striations. So knowing how to draw a sarcomere is going to be super important for this chapter. So be able to draw it and be able to articulate it from scratch with what is happening. But that is our thick and our thin filaments, our myofibrils, and all of those pieces. This is interesting, and why this really matters is when we're looking at some disorders of the skeletal muscle system. One of those is going to be muscular dystrophy. If you've ever heard of muscular dystrophy. So this is a common muscle disorder or muscular system disorder. And what it is it is going to be a muscle destroying disease is what it's considered. So what is happening is that with this particular type of disorder, it is going to be a defective gene for the protein that links the thin filaments to the extracellular matrix and stabilizes the sarcolemma. So basically, we're now going back to our thin filaments in our sarcomere, and muscular dystrophy is actually destroying the protein that holds those in place. So that is a challenge because it is a usually some sort of recessive genetic condition. So if you've got the genes for it and it is recessive, you typically will show up and have it, and it is a degenerative disorder. So you can see how this is a sweet little boy with muscular dystrophy, and it's going to affect his skeletal muscle tissues. Okay, so all of what we've just talked about was under the category of myofibrils. So way back up, this is a heavy chapter, way back up we said that there were three different modified organelles that are in our sarcoplasm. We just spent all that time and we only talked about our myofibrils. We now have to talk about the other two organelles, sarcoplasmic reticulum and T tubules. So when we're looking at our sarcoplasmic reticulum, this is the second modified organelle in our skeletal muscle tissue. So I'm just going to call it our SR, our sarcoplasmic reticulum. So this is a cool feature. This is a network of ER. So it is a network of smooth endoplasmic reticulum that is going to be specific to the skeletal muscle fibers, so it's called sarcoplasmic. All of these are going to run longitudinally along the cell, and what they're actually going to have is something that is going to be called a terminal cistern. So the way this sarcoplasmic reticulum works is these are intracellular tubules. And these intracellular tubules are found inside the cell, and they're going to regulate muscle contraction. So as they regulate muscle contraction, they are doing it from the inside of the cell. It's a very cool. So these are going to surround each myofibril. And they're going to surround each myofibril at the H zone. So they're going to essentially, in a nutshell, a better way to think about it is they're going to form a cross bridge at the I and A band junction. So whenever we have all the way down our muscle fiber, we're going to have the striations of the dark band and the light band that looks like this. What you're going to have is you're going to have your T tubule. Let's see if we have a picture. Yeah, here's our I band and here's our A band. Our T tubule is actually going to be going down that section. So you can see how this guy right here is going to be coming down this I band. He's at a cross section. So that means our sarcoplasmic reticulum is going to form bands down right here. A cross bridge at the I and the A band junction. What the sarcoplasmic reticulum is responsible for doing, and this is super important, is here's responsible for regulating your calcium levels. Calcium is going to be super, we talked about it in bones, but it's going to be super important when we get to skeletal muscle. So how much calcium you have is related to how much your skeletal muscles are able to work. Your sarcoplasmic reticulum regulates your calcium levels. And the way it does it is it will actually store calcium, and then it will release the calcium on demand. So when calcium is needed, it will release it. The way it's going to do this is it's going to store these in the structure called a terminal cistern. So cistern is essentially a fancy word for a jug or some sort of vase that will hold liquid. So a terminal cistern is going to be the final resting place of calcium, and this is going to be found inside the cell because it's intracellular, And these are going to be tubules that hold calcium and release when needed. Okay, so that's your sarcoplasmic reticulum. The next one is going to be your T-tubules. So this was our third, this guy right here. This was our third modified organelle, was a T-tubule. And so what's happening with this is a T-tubule is going to be also at the I-A band junction. So at the I and the A band junction, the T-tubule will be made up of the sarcolemma. So that the plasma membrane will jut into the cell, and it will form a tube. So this in a traditional surface area would be my sarcoplasma, my sarcolemma. It's essentially my plasma membrane. It's the surface of the cell. But what we mean when it juts into the cell is it will actually do this, and it will form a tube. This is your T-tubule. So when it juts into the cell, it essentially is a closed off bubble of sarcolemma that is jutting into the cell. So this T-tubule is formed by the protrusion of the sarcolemma into the cell's interior. The reason we have this is it will actually increase the surface of that plasma membrane or the surface of that sarcolemma greatly. So that's very, very, very important. And it will allow electrical nerve transmissions to reach deep inside the cell. So if we are going to have any kind of electrical change on the outside of the cell, guess what you can also do is you can now have that inside the cell. So this is what I meant when I said we're going to revisit some topics and concepts from chapter 3, the whole how the plasma membrane works. The sarcolemma, which is the plasma membrane, works that exact same way. So that is what we're talking about. We can increase our surface area of our sarcolemma, we can put it inside our cell, and we can have electrical activity deep inside our cell. Okay, so now with our T-tubules and our SR, they were both occurring at the IA band. So that's really cool. They actually both occur at the IA band in such a way that you can actually put them together. And this is going to be a triad. So a triad is actually going to be a T-tubule. And it will be on either side, it will be a sarcoplasmic terminal cistern. So a triad means 3, and so we'll have this T-tubule, and then on either side, we're going to have our sarcoplasmic, our terminal cistern. So if we're looking at this example right here, our T-tubule, remember, is jutting into it. Our sarcoplasmic reticulum is actually now going to be on either side. So here is my terminal cistern for my sarcoplasmic reticulum, and then here is my other terminal cistern for my sarcoplasmic reticulum. You'll notice if you put them together, you have 3. That is where you get the triad from. This is inside the cell. So we can have our plasma membrane or our sarcolemma give electrical activity deep inside the cell, and our sarcoplasmic reticulum and our terminal cistern is going to be where we actually store our calcium to release it inside the cell when needed. So to put it all into perspective, that is what is happening. So with this triad, it's our T-tubule that is protruding into the space. And this T-tubule will come together with our sarcoplasmic reticulum cistern membranes that also protrude into the intermembrane space. And these are going to control the opening of our calcium channels. That is exactly what we just drew right here. So the whole reason we have these triads is when we're actually going to have a change in our electrical impulse. This whole process will eventually go through these steps that we're going to talk about a little bit later and release calcium. We need calcium inside our muscle fiber in order to contract. A triad with the T-tubule and the two terminal cisterns of the sarcoplasmic reticulum allow that to happen. So to kind of look at what is going on with these sections, you can see we already briefly talked. Here's our I band and our A band. So remember, kind of see where those are. Here's our I, here's our A. And then write down the connection of the I and A bands. So right here, this is where you're going to have your T-tubule and your terminal cisterns put together. We have 1, 2, 3. There is our triad. And that will be along the entire muscle fiber, all the way every time you have an I band or an A band. So that's what's going on with that section. So then, how we actually use our sarcomere. This is going to be called the sliding filament of contraction. And the way this is actually going to work is we are contracting our sarcomere. So when you contract the sarcomere, you shorten the sarcomere. So if I have, here's my muscle fiber, and this is a Z disc, Z disc, Z disc, Z disc, Z disc. And this is a sarcomere, 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 all the way down. We are contracting the sarcomere. And the way you will actually do that is 9.4. When you contract it, what you actually are going to get is, here's my first sarcomere, but here's my second. That's a sarcomere. And here's my Z disc and my Z disc, sarcomere, sarcomere. So we have essentially now contracted this guy all the way down to this distance. So from here to here. We've shortened it, and you do that by contracting the sarcomere. And the way that's going to work is called cross bridges. So we're actually going to generate force by creating cross bridges. And it's going to be involving our thin filaments mainly. So we're going to go back to our actin and our myosin real quick. So when we are super, super relaxed in our sarcomere, our thick and our thin filaments overlap only slightly at the ends of the A band. So I'm going to drive all the way back up to our sarcomere. Our thin filaments and our thick filaments only slightly overlap. So what we're talking about is the section that is, find a color we have not used, here and here. That is the only section where you will actually have your thick and your thin. You'll notice in the middle that there's no overlap, and you'll notice here that there's no overlap. So this section of, let me erase that so it doesn't show up on the notes. So with this overlap of our thick and our thin, that is where we're going to have our sarcomere shorten. Let me drive down. Okay, so when you overlap slightly at your thick and your thin filaments, the sliding filament model of contraction states that what will actually happen when you shorten your sarcomere is that the thin filaments will slide past the thick filaments, causing actin and myosin to overlap more. So because it's just easier to draw, here's my sarcomere. Here's my thin, 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 thin. Here is my thick. Just to make it easy, we're not going to draw the whole 9 yards. But remember, he does not extend all the way to the end, and he does not touch the thin filament. But holding this in place was going to be our small little protein that was holding this in place, that was attached to the end of our thick filament. Okay, so this is the world we're living in. What we mean with the sliding filament of contraction is right here. This is the end point for our thin filaments. Right here, that's where the green ends, and right here. All of this is essentially open space. And what we're saying is when we have the sliding filament model of contraction, thin filaments slide past thick, causing them to overlap. So you're going to move your Z disc towards the middle. You're going to move this Z disc towards the middle. And you're going to take right here this line of actin that is not super overlapped at this point, but right here. And you are going to make him overlap with the myosin much, much more. So if we were to draw that, we now have taken this one sarcomere. We're going to do this one because he's been consistent. And we have moved this guy this way. So when we actually do that, we have not shortened, here's our actin. We have not shortened our actin at all. We have just moved this one and this one closer together. We have also not shortened our myosin at all. So he is still going to be as thick as he was. He is just now more overlap between your actin and your myosin. So now we have this as our overlap between our actin and our myosin. That is much, much different. The big premise here is that you have not changed the length of the thick or the thin filament. So with the sliding filament of muscle contraction, neither thick nor thin myofilaments contract or shorten. You are not shortening the myofilaments. You are shortening the space that they overlap. So this is important. You are not actually shortening any structures. You are essentially only decreasing their space apart. So you'll actually have these cross bridges form and break several times where you pull the thin filament closer to the sarcomere in a ratcheting action, which causes the muscle fiber to contract. So we will have our Z disc, which are these guys. They are pulled toward the M line, which was this guy. So that makes sense. Our I band will actually shorten because that's our middle line right here, where it's only thick filament. Our Z discs will become closer, which makes sense. Our H zones will disappear, which is going to be that space in the middle. And our A bands are going to move closer together. So here is a great picture. So here is our sarcomere. So we are doing Z disc to Z disc. So this is our sarcomere. You'll notice that here is my actin, which I will highlight in yellow. And then here is your myosin, which is going to be in the red. So it's this structure right here. You're going to pull the Z disc toward the middle. And when you pull the Z disc toward the middle, you are essentially now going to have a sarcomere that is only this wide. And with that, you're not going to have any space in the middle. So I still have the thickness of my thick filament, and you'll still have the length of the thin filament, but you'll notice that there is essentially massive overlapping. Whereas this space was here before, there is now none of that space right there. You can see it here where it's got the dark space, the A band, or now here is our A band. It has drastically decreased. So that is actually our sliding filament model of contraction. You're essentially going to make the sarcomere smaller. You're going to contract it. Okay, that blends into the premise of 9.4. So 9.4 is actually going to be how we go through the process of contracting all of our sarcomeres. So 9.3 and the sarcomere is the first thing that's important in this chapter. The second thing is going to be the entire section of 9.4. And this is going to be a muscle fiber contraction, how you actually shorten the sarcomere. So in order to have this whole process happen, it is going to be activated by the brain, and it's going to be triggering a signal from the brain. So we're going to start here. It's going to go down the spinal cord, and then it's going to go out to your actual muscle. It's going to activate these muscle fibers. The communication from the brain all the way down takes the form of something called an action potential. So we're going to talk about voltage again. But when you all get all the way down to the end and we've got this communication happening with our muscle, there is a specific communication pattern that will actually happen, and that will be only done through the neurotransmitter acetylcholine. So when we get down to the connection between the nerve and the muscle, your neurotransmitter that will occur in the skeletal muscle system and how this whole process works will only be acetylcholine, which is ACH, or acetylcholine. That is what we will be talking about when we're talking about neurotransmitters and the connection between the nervous system and the muscles. Okay, so this is a 4-step process that we're going to talk about real quick. We're not going to talk about our chemically gated channels. Okay. This is where we're going to go. Okay, so looking at our 4-step process, we are going to start with activating our skeletal muscle. We're going to generate an action potential, and we're going to move it through our entire system. So this is going to be a 4-phase process. So my notes are going to kind of jump around a little bit in the PowerPoints, but we're going to marry everything up at some point. So a muscle fiber contraction is actually going to occur in 2 phases. The first phase will occur and be phase 1, and it's actually going to be the motor neuron. And the motor neuron is going to stimulate the muscle fiber. So this first phase 1, we're going to have the motor neuron stimulate the muscle fiber. This is going to have a part A and a part B. So the first part A is going to be the events at the neuromuscular junction. We're going to talk about that one. And then after you have the events at the neuromuscular junction, you're then going to have a generation of an action potential, which is that communication in the nervous system, across the sarcolemma. So phase 1 is the motor neuron, the nervous system, connecting to the muscle fiber. That's going to happen because there's going to be part 1, which is events at the neuromuscular junction, and part 2, an action potential all the way down the plasma membrane, otherwise known as the sarcolemma. And then phase 2 of a skeletal muscle contraction will be excitation contraction. So now we're going to couple. Part A is we are actually going to couple our pieces together, and this is going to be through our excitation contraction. And then after we do this step, we are going to go through cross-bridge cycling. And when we finish our cross-bridge cycling, we will have gone through the entire skeletal muscle contraction. So what we're getting ready to do is go through 4 steps: neuromuscular junction, action potential, and then that's going to do excitation contraction, and then we're going to do cross-bridge cycling. So these 4 steps are essentially going to be what causes the communication from the brain to cause your skeletal muscle to contract. So we're going to go through all of these steps. Okay, so we're going to first talk about the part 1, these events at the neuromuscular junction. So when we're talking about the NMJ is how you will hear it a lot. This is the neuromuscular junction. This is the connection between the nervous system, neuro, and the muscular system, muscular. So the connection point between a nerve and a muscle is the neuromuscular junction. And at the neuromuscular junction, what you'll actually see is this will be the, here's my neuron, and it will be called an axon. It's the final communication pathway. So it'll be the axon of a motor neuron, and it will be connecting to what is called the, let's do this as a different color, the motor end plate of a muscle fiber. So we're going to have the connection between the brain and the nervous system. Here's our little brain. That's a bad one. Let's try that again. Woo. And our actual muscle. So here we're sending a communication down the axon of the motor neuron, and where they actually connect right here is considered the junction. That's where that came from, of the neuromuscular junction. And it's the axon of the motor neuron and the end plate of the muscle fiber. So this is what we're talking about with this neuromuscular junction. So you can see here how we have the muscle axon coming down, and he connects to your muscle fibers. The way they actually connect is going to be super specific. So here is my lovely little communication down. Here is my sarcolemma. The way they actually communicate is there's going to be little branches that come off that will have places that will attach and communicate, little buds. And they will not actually connect. So there will be a space between the end point of the neuron and the actual muscle fiber. This is going to be the synaptic cleft. So the space is the synaptic cleft, and this will occur for all of them. But what we're doing with this, if this is our axon coming down, this right here is our last place we can be. So it's our axon terminal. Our axon terminal then has the synaptic cleft. And if we need to get communication across this pathway, we need to get communication from the neuron to the actual muscle fiber. The way that happens is through something called membrane-bound synaptic vesicles. So across this lovely little cleft right here, we will essentially send something from our axon terminal all the way across to our plasma membrane, our sarcolemma. So that will release tiny little vesicles that will float across, and then they will bind to the other side. Inside these vesicles will actually be our communication. So they will have inside, not that that shows up, our neurotransmitter. So we will actually have these synaptic vesicles with the neurotransmitter acetylcholine, and they will cross from the axon terminal, cross the synaptic cleft, and then bind on the sarcolemma. So that's what we're doing here. Here is a great blown up example of what we're talking about. So here is our axon terminal. Kind of looks like here's the buds. Coming out of it and across is going to be our synaptic vessels. Inside our synaptic vessels are our acetylcholine neurotransmitters. So these guys are going to be found inside. As they travel across the synaptic cleft, they are going to bind on the other side of our sarcolemma. So this is what is happening with this connection between the nervous system and the muscular system. So how that actually works, here is our 4-step process. I like to describe it up front that we are talking about. Okay, so with this 4-step process, we've talked about the neuromuscular junction. Now let's see how that works. So that is going to be these steps right here. So the events at the neuromuscular junction are Phase 1, Part A. The events at the neuromuscular junction. How do we get communication? First step, you are going to have an action potential, which is a communication, an electrical change, will arrive at the axon terminal. Next step is voltage gated, meaning it is closed and the only thing that opens it is going to be a change in voltage. So a voltage gated channel is specifically going to be calcium, and these will open. If I open a voltage gated calcium channel, what I'm going to have is calcium will enter the neuron. So that is going to be a 3-step process. We have an action potential arriving at our axon terminal. Our voltage gated calcium channels will open. Calcium will enter this neuron. When this happens, this will cause the release of our ACH. So this step sequence is going like this. As we release our ACH, it will cross the synaptic cleft. And as it crosses the synaptic cleft, it then is going to get to the other side and it will bind to ACH receptors on the sarcolemma. So all the way across the sarcolemma, the synaptic cleft on this muscle cell, we are going to bind our ACH to receptors that like ACH. That's what that means. And after our ACH binds, it will open sodium channels. What we know about sodium is that it is a high concentration outside of the cell. We know that to be true. If we open sodium, this was chapter 3, then sodium will enter. If we have sodium entering the cell or entering the muscle, now what we're going to have is essentially a change in an end plate potential, but we're going to change the resting membrane potential of the sarcolemma. So that is a very complex process that will then is essentially a multi-tiered process. But I'm going to come on down to this guy and then we're going to blow him up and look at him. So this is exactly what is going on. So we've talked through 1 through 6. We've written them out, but we're going to just talk through them right now. So here is my axon. Here is my skeletal muscle right here. So this is the connection between the two. So what is happening is, number 1, an action potential arrives at the terminal membrane. He comes down. Number 2, voltage-gated calcium channels are going to open. That means calcium will then enter the cell down its concentration gradient. Calcium entry into the cell will cause acetylcholine to be released in the synaptic vesicles. ACH will diffuse across the plasma membrane and then bind to receptors on the plasma membrane or the sarcolemma. ACH will open sodium channels that will allow for sodium to enter the cell. Sodium will go in. When we have sodium going out, you will have some potassium, but what I want you to focus on here is more sodium ions will enter. So we're going to focus on sodium. So as the sodium enters into the cell, it will essentially cause an end plate potential. You will change the plasma membrane concentration of ions, therefore you'll change the actual measurement, the potential, you'll change the charge. So this is what we're talking about. It doesn't seem that super complicated, but it is going from the neuron to the actual muscle. This is the process that works. So that's the first step, the events at the neuromuscular junction. Okay, this second event we're going to talk about is after we get, so this is going to be part B. After we get the action potential down our axon and we have our neuromuscular junction, now we're actually going to have an action potential down the sarcolemma. I don't think I have enough room. And he will travel down the sarcolemma. What we mean by action potential traveling down the sarcolemma is if we have, let's draw it down here. If we have our neuron connecting here and we have our muscle fiber here, everything that we've just talked about happens here, neuromuscular junction. Now what we need to do is have it go this way and we need to have it go this way, because remember, our muscle fiber cell that we've talked about a lot is a long, narrow muscle fiber. So we need to not just have the actions occur at the actual junction, we need to have it travel down the sarcolemma. So that's what this process in this part B is really talking about. So the resting sarcolemma is polarized, meaning there is a voltage across the membrane. So the inside is negative compared to the outside. We know this to be true because this is what we talked about in chapter 3. The inside is negative compared to the outside. And this will occur in 3 steps. But what we're going to do is a series of electrical changes. So with this action potential, we're going to get a series of electrical changes. So it's a 3-step process. Step 1, you will have the end plate potential. So this is ending at the very end of step part A. So when we had events of the neuromuscular junction right here, we ended with the change at the very, very end right here, and it ended in an end plate potential, or it ended in the change in the end plate potential. That is how this whole thing ended. So now how we're starting the next one is we're going to start with an end plate potential. So at this point, we have our ACH, and the ACH will bind to our sarcolemma. And when it binds to the receptors in the sarcolemma, we've already talked about this because it's the first couple of steps, or the first couple of steps ending part A. It will open ion channels, And if it opens these ion channels, sodium will move into the cell. Sodium will move into, again, we've already talked about all of these pieces because it is a sequence of steps, so they build upon each other. Now here we go. When sodium moves into the cell, the inside will become less negative, or you can call it more positive. But if we have essentially now changed, here's our, remember these guys, here's our out, here's our in. We are talking about sodium going into the cell. That means that we are adding positives. So you are going to be less negative. So if this was negative 60, negative 65, essentially this is now going to become negative 60. We're going to add positives. But the numbers were just as an example. This change is going to be called a local depolarization. So the local depolarization is this change. That is also called the end plate potential. So we have done the neuromuscular junction, and now we have caused an end plate potential to occur. And remember, all we're really doing is traveling down the rest of the muscle fiber. Okay, so that was end plate potential. The second part is going to be continued depolarization. So continued depolarization. So as we have our end plate potential at our neuromuscular junction, so we're talking about this lovely connection from our neuron to our cell. We have done everything right there. But now what's going to happen is we're going to depolarize. So if the end plate potential causes enough change in the voltage to reach a threshold, voltage-gated sodium channels in the membrane will open. So if all of that will happen right there at the neuromuscular junction, and we're going to have sodium going in the cell here, that will then trigger the next ones to have opening sodium. And if we open the sodium, guess where sodium will go? And then that will travel further down, and then we'll open sodium. And if we open sodium, sodium will go in. So this depolarization will travel down the length of the muscle fiber. And it will be due to a large influx of sodium that is unstoppable. And it will cause a muscle fiber contraction. And this essentially spreads the action potential down the sarcolemma from one voltage-gated sodium area connected to the next, so everything depolarizes. And then down the length of the entire muscle fiber or the entire straw, you have the influx of sodium. That is the depolarization of the action potential. And then that leads us to the next part, which is part 3, repolarization. After we have opened up all of the sodium channels, we now need to close them. So we need to go back to baseline. So we need to go back to our baseline. The way we do this is we are going to repolarize. So we are going to close our sodium channels. If we close our sodium channels, no more sodium will enter. But what we're also going to do is we're going to open calcium channels. And calcium will move out of the cell. So if this is my sarcolemma right here, and this is outside the cell, and this is inside, we are saying that we are now taking our sodium and we're closing, so it doesn't go out and doesn't go in the cell, but we're going to open our calcium, which means calcium will go this way. calcium will go out of the cell. If you do this, then you are essentially going to give that take that resting membrane potential back to its normal initial resting state. So this is what's happening with our repolarization. It is due to our sodium channels closing and our potassium channels opening, and the movement of our potassium ions takes our resting membrane potential back to normal. So that is part one, part A and B of phase one. But here is an action potential graph that we are going to spend a lot of time on as we go throughout this course. So this is going to be time down here. It's done in milliseconds. So just start is going to be here, end is going to be here. And then this is going to be membrane potential. And this is measured in millivolts. So here's where 0 is going to be, all the way down to negative 90 millivolts and up to positive 30. So remember, the inside of the cell is actually negative. So we're down here. And then what will happen is you will open your sodium channels, and you are having an influx of a positive ion, which means you are becoming less negative. So that's what this means. When you open the sodium channels, you become less negative. When you reach this positive 30, you're going to close your sodium channels, and you're going to open your potassium channels. When you do that, potassium is going to enter the cell, becoming more negative. And then once you get down to about right here at some time frame, right here, you will actually have your potassium close. Your potassium channels will close. So this is kind of what's happening with our sodium-potassium resting membrane potential. You do need to spend a lot of time and know this process and what this graph looks like and be able to interpret it, because it's going to get more complex as we go. Okay, that was phase one. Now we only have the last two parts. We have phase two to do, which was excitation, contraction, coupling. So we have communicated from our neuron down to our muscle through the neuromuscular junction. And then we have communicated across our sarcolemma, across our muscle fiber, that we now have a change in our membrane potential. So the next two processes are going to be involved with making our sarcomere contract. So this was our phase two. But this is going to be part A. Excitation, contraction, coupling. So here what we are doing is we are going to be connecting deep inside our muscle fiber. So excitation, contraction, coupling. This is where we are taking our action potential, the events that have come from coupling our action potential along the sarcolemma, and we are going to couple it with the sliding filaments of contraction that we talked about in the first couple of sections. So we're going to combine the action potential with the sliding filaments. That is what's happening in this process right here. So this is going to be deep inside the cell, and it's going to involve our sarcolemma, our T-tubules, our calcium, and our sarcoplasmic reticulum. So we're going to kind of briefly go through all of those pieces. So I'm going to jump down to the image where we have them all. Okay, so with our excitation, contraction, coupling along the myofilaments, what is actually happening in this first step? Let me find my notes, I got ahead of it. With excitation, contraction, coupling, we are going to have the action potential will travel down our sarcolemma. And as it's traveling down the sarcolemma, it will also travel into the T tubules. So remember, our T tubules was our sarcolemma that did this, so it could dive deep inside the cell. So if we have a action potential traveling down our sarcolemma, it can also then cause these changes deep inside the cell. So it'll travel to the sarcolemma and it'll travel into the T tubules. Now what it's going to do is the next step is it is actually going to cause voltage sensitive proteins. Let me shrink that a little bit. It'll cause voltage sensitive tubular or tubule proteins to change. And when these proteins change shape, they will open calcium channels. When they open calcium channels, what they will do is they will open these in the terminal cistern of the sarcoplasmic reticulum. So to stop right here, remember on either side was our terminal cisterns of our sarcoplasmic reticulum. This is a very heavy chapter. Our action potential has traveled down our sarcolemma. It travels into our T tubules. Inside our sarcoplasmic reticulum, we have calcium. As the change in voltage triggers our calcium channels to open, it will then cause our terminal cisterns to release our calcium. And when that happens, we will essentially have calcium will flow into the cytosol or into the cytoplasm, sarcoplasm. Now we are inside and we have free calcium floating around. Now we're going to go uber detailed. This calcium that is now floating around inside the cell, calcium will bind to troponin. Do you remember what troponin was? Troponin was the protein that held our thin filament in place. So if this was our thin filament, and this was our actin protein, we are talking about the tropomyosin is the green string, or the green string is held in place by our tropomyosin, which was these lovely little stars. So the calcium will bind to these little places, will bind to our troponin. So calcium will bind right there. And when calcium will bind to troponin, it will remove the blocking protein. So it binds to troponin and then it will move troponin. And it will move the troponin or the blocking protein off the tropomyosin, which was the green string that the actin filaments were attached to. So basically, calcium will bind to troponin. Troponin will move, and it takes the blocking protein off of tropomyosin. And as it does that, it essentially opens tropomyosin. And if we have this blocking protein removed, then what we have essentially done is we have exposed the active binding sites for our myosin. So we have exposed the active binding sites for our myosin. and that means we're exposing it on our actin. So I'm just going to actually say that a little bit different. Exposed the active binding sites on our actin filament to bind myosin. So this is what is happening for our excitation contraction coupling. And it's called excitation contraction because we've got calcium moving, so it's being excited, and we're coupling because we're going to essentially connect our actin and our myosin. So we're going to zoom in on these images real quick. Yeah, it's a little fuzzy, but we're going to do it. Okay, so the actin, the action potential is traveling down the plasma membrane of the sarcolemma, and it's going into the T tubules. So it's coming down and into. When it comes down and into, it's going to cause calcium to be released from the terminal cistern. Calcium will be released into the actual just cytoplasm. So now we have calcium floating around inside the cytosol. Then we get to this. So here is our actin. Remember, our troponin was the little thing that was holding it in place. So that's this guy right here. So our calcium will bind to our troponin. That's what's happening right here. And when it binds to our troponin, it moves it and it will open up the spots. So you'll notice on these guys right here, all of these, all the little dots are blocked. But if you move the string, you can see how all the little dots are now open. Those dots are where we're going to form a connection between a myosin and an actin filament. So in Mastering A&P, there's actually a couple images of this that are really cool to help kind of show what's going on. And then this video right here in the PowerPoints that are posted is also great to watch. But what we've done is we've had the action potential travel down the sarcolemma. It's opened calcium channels. Calcium has flowed into the cell. Once it's in there, it binds to troponin. It removes the protein, so tropomyosin is gone, and it exposed the active binding sites. So now, if we have exposed active binding sites, you are actually going to have myosin will then bind to the actin and form a cross bridge. So if my myosin, my thick filament looked like this with my golf club heads, and my thin filament was going to have this guy with my molecules of actin now available. I'm going to do it as an open because my binding sites are now available. We are going to connect our actin and our myosin. So your myosin head will connect to the actin filament. So it'll form a cross bridge. So the myosin head will connect to the actual actin filament. So now you have the cross bridge, which is myosin and actin connected together. So when you have no release of calcium, so when you've got low intracellular calcium, normal, this is normal, no muscle activity. Your tropomyosin will be in place and it will block the actin sites, and the myosin heads cannot connect. So the muscle fiber is relaxed. This is normal, no activity, no contraction. The only reason we'll have contraction is because we will change the calcium concentration. So now that we've formed a cross bridge, we're going to go through part B, which was cross bridge cycling. And this is the last concept for this section. cross-bridge cycling. So here's just the review of our cross-bridge. Calcium becomes high in the cell. Calcium will bind to troponin. Troponin will change shape and move tropomyosin away from the binding site. The myosin head will bind to actin, forming a cross bridge. And then we get into the last two steps, which is the cross bridge cycling. We are going to cause this cross bridge to form multiple times. So you will form a cross bridge, then you will break a cross bridge. So cross bridge, remember, is actin and myosin connecting. So you'll connect actin and myosin, then you'll break actin and myosin. Then you'll connect actin and myosin, and then you'll break. And then you'll connect, and then you'll break. And you will cycle through this process. That's why it's called cross bridge cycling. And this is going to occur until calcium leaves the cell. So there are 4 steps to cross bridge cycling. The breaking and forming of these processes. So, cross bridge cycling, 4 steps. The first step is you will actually have formation of the cross bridge. So, cross bridge formation. You will make the connection between actin and myosin. So, this will be with the presence of ADP plus a phosphate. So you are going to go back to the sections of early Unit 1 where we were talking about ATP, and it had energy if you broke the terminal phosphate and made ADP, adenosine diphosphate. We're going to revisit that concept now. So with a cross-bridge formation, we're doing it with ADP and phosphate, so an adenosine diphosphate. So what's happening here is the myosin head is attached to actin. And this is going to form a cross bridge. Cool. Well, we already know that to kind of be true. So then the next step is going to be 2. So your myosin head will bend. And when your myosin head will bend, your ADP and your phosphate will release. So when we're talking about our myosin head, what we're talking about is, here's my, oh, that's not the one I want. Let's try this. So here's my myosin head. You'll notice that I've drawn them all kind of at 90 degrees. So what we mean is your myosin head will bend is that it will go from 90 degrees to something that's a little bit more, less than 90. So let's say 45. So we'll take it and our heads will look like this. We will essentially have bent the heads this way. So you have decreased the angle of the joint. So your myosin head will bend. And when it does this, it will actually lose ADP and your phosphate. It'll pop off. So then what it will do is if the ADP and phosphate has popped off, then it will pull, we're not quite to part 3 yet. If the myosin head bends, it will pull actin toward the M line, toward the middle. So it'll pull actin towards the M line. And then what it will do is it will go through a process of cross-bridge detachment. Cross-bridge detachment. And this will be with ATP. So now you will add ATP. So ATP will attach to myosin. And when ATP attaches to myosin, it'll break the link between your actin and your myosin. So they've been connected, they've formed a cross bridge. When you add ATP, it breaks the connection. And it will do this because the myosin head will detach. So you will break the cross bridge. And so now we are not connecting your actin or your myosin. So then we go to step 4. Is now we have our myosin head in our bent position. We need to put it back. So this will be the cocking of the myosin head is what it's called. So as we put the myosin head back in place, we move it back to its 90-degree angle. So we cock the myosin head back. What will happen here is your ATP will hydrolyze, and it will go from ATP to ADP plus that phosphate. So we'll have our ATP hydrolyzed to adenosine diphosphate, and we will remove our myosin head back to normal. So we will go from where we were bent a little earlier in step 2, where our heads were like this. So here's our myosin head. We will now go back and put them back at that 90-degree angle, and we're going to do it like this. So that is what is happening with this step, but we're doing it with ADP hydrolyzing. So when we do it like this, and we've got our ADP and our phosphate, we are then back at ADP and our phosphate. We are back at step 1. So cross bridge cycling is the key word. It is a cycle. So you're going to go through all 4 steps. So I'm going to blow up and talk through this guy right here. This is a big one that you need to know. If you can draw this, then you know it very, very well. So we're going to start with the first one is forming the cross bridge. You'll notice that this is with ADP plus phosphate, and it is going to be connected myosin plus our actin. You can see how they're connected. Then the second step is you will bend the myosin. So you can see how now we've changed that angle. So it's bent the myosin head. So you lose ADP plus the phosphate. And you have pulled the actin. So you can see how here he's being pulled this way. You'll notice this way is this way, and the middle is the M line. So you're pulling it towards the midline. Essentially what you're doing is you're taking the golf club head, you're attaching it to the thin filament, you're moving the golf club head so it pulls everything towards the middle. Then in step 3, you're actually going to add ATP, and you will essentially break myosin plus actin connection. So that's what's happening in 3. And then when you get to number 4, you will put your myosin head back to normal, and you will then have a DP plus the phosphate. So your ATP will hydrolyze, and you put your ATP will hydrolyze, and you put your myosin head back to position, so it is ready to then connect. So this is a cycle. So it will form here, it will break here, it will form another one, it will break, it will form, it will break. But as it does this, it will slowly move things toward the middle. So you are sliding these filaments over each other, that sliding filament of contraction. Okay, the important thing that's really, really cool to talk about with this is the concept of rigor mortis. So one thing that's cool about rigor mortis is if you've ever followed any clinical, medical, pathology-driven TV show, you've probably already talked or heard about rigor mortis. So this is actually a really cool concept. So rigor mortis is how we know that cross-bridge cycling works. So this is a concept. It's not a disorder. It's not a disease. It's more of a characteristic. So 3 to 4 hours after death, you will have your muscles start to stiffen, and they will hit peak stiffening or peak rigidity about 12 hours after post-mortem or after you've died. So what is happening with rigor mortis is, if you think about it, you are essentially, this is going to be after death. It doesn't cause death. It is a characteristic after death, but it shows the role of ATP. ATP you get by breathing, going through cellular respiration, having your mitochondria do what they should be doing. You have ATP happening when you are alive. If you are not alive, you do not have ATP being synthesized. So rigor mortis is directly related to ATP not happening. So what you'll actually do with rigor mortis is you will have a cross bridge form. A cross bridge is the connection between your actin and your myosin, and that causes this muscle rigidity that you see. However, if you have the cross bridge form, there is no breathing, so therefore there is no ATP, therefore there is no cross bridge breaking. So if you do not ever break the cross bridge, you are left with the actin connected to your myosin, and there is no break. That is where you get this rigidity, because you need ATP for cross bridge detachment. So if you do not have that, you are going to have myosin staying connected to actin, causing a constant state of contraction. So essentially, up here in this process, you will stop at step 2. So you will stop essentially at step 2 in rigor mortis, because you can't add ATP if you're no longer alive. So this whole process doesn't cycle. You just stop. You will eventually see this rigidity disappear. So it does hit peak about 12 hours, but then after that, it will start decreasing the contractions. Your muscles will stop being contracted. And the reason it'll do that, it's not ATP and it's not breathing and it's nothing else, simply because as your proteins will start to break down. Actin and myosin are both proteins. So if you 12 hours after death, your body will actually start to decay. Proteins will start to decay. Actin and myosin will detach because they've decayed. It's not because they've actually decreased and broken the cross bridge. So that's a really cool way that with rigor mortis, we know that ATP is important for this cross bridge cycling, because without ATP, the process completely and utterly stops. Okay, so that's 9.1 to 9.4. As I said, they are very, very heavy concepts, and it's only going to get more complex for 9.5 to the rest of the chapter. So the focus for this is understand 9.1, understand 9.2, but dive in deep to 9.3 and 9.4. The sarcomere, be able to draw it, know what all of these bands and lines are, how actin and myosin are connected, what proteins make them up. You can't understand anything from 9.4 on how a skeletal muscle contraction occurs if you do not understand the structure. So 9.3 is the structure, 9.4 is the function. So those are the focus for this first part, or this week, of chapter 9.